I'm going to talk about surveillance of the food supply using whole genome sequencing, and I really appreciate the prior presentations that have really helped set up, so I'm not explaining what a SNP is. So my thanks to Dr. Engelthaler and others. So at FDA, a big role of outbreak investigation is traceback. We want to first preclude further distribution of the product and further illnesses. And then also the information from outbreak investigations and traceback to find the source of an outbreak provides very useful information on practices that could lead to a future outbreak. And, but these are very complicated investigations, and some of the complexity of these investigations is related to the complexity of modern food production. You have foods that are produced from multiple components that come from all over the world. Spices and produce are shipped, all over. You have different processing and distribution, so very complicated pathways for food that makes ultimately traceback very difficult and might end an investigation in its tracks. We have new food processing technologies and that are leading to a new understanding of pathogen product pairs, like pathogens that are associated with different products. And we're seeing, especially at FDA, with Listeria and the association of Listeria with new products, including fresh produce that has occurred in recent years. We know that pathogens have a diverse and complicated ecology, that pathogens such as Salmonella have a free-living existence in the environment and can move from surface to surface. And so the association of Salmonella enteritis with eggs is a very time-worn rule, but we know that enteritis could come off those eggs and into other products. So we have to be aware of that. We also are faced with the reality that inspectors cannot be at every establishment at every time. And so we have to make the best use of our regulatory resources. And so, that's going to hinder foodborne traceback investigations. And lastly, the source tracking tools that we've used at times lack the resolution or robustness and the rapidity to be of maximal use during a traceback investigation. So these are some reasons why we should develop and why we are developing a whole genome sequence-based surveillance of the food supply. Whole genome sequence, as was discussed earlier, is statistically robust, exact data. So these are not restriction fragment length polymorphisms. They are nucleotide sequences, and we can compare and determine if they were in the same position, if they have been changed. We can reconstruct the phylogeny, the evolutionary relationship in a set of data. So that provides very robust and improved specificity related to the connection of isolates in a food supply and clinical isolates. And then from a practical standpoint, whole genome sequence, the development of whole genome sequence assays are really agnostic to the target pathogen, so you can develop workflows that would cover many bacterial, fungal, viral pathogens. And then there's many ways that you can take a data set and analyze it, as we've seen in the prior presentations, to learn not just about the phylogenetic relationships, but also the genes that it carries, the likelihood that it encodes virulence, antibiotic resistance, so on and so forth. The data is easily stored and transferred as a digital form, and as we move forward, the cost and the availability of data is increased. So the dropping costs and improved throughput amount of data coming out of these out of whole genome sequencing. And lastly, there's a potential for analyzing non-cultured samples, or so-called metagenomic samples, and that is an area of great interest. On the other hand, and this would really apply to all microbial source tracking methods. When we think about food sampling, there are some limitations in terms of the forensic and epidemiologic value. So we have to remember that we're looking at organisms that are on a product, and the ability to detect those organisms is dependent on the sampling. And you know, the pathogens are usually present at very low levels, and sampling schemes have only a certain have a limited ability to detect the pathogen in a particular product. We also have to be thinking about the outbreaks that involve more than one subtype and how we interpret that. So, so there's been examples in the published in the literature and what we're seeing in FDA where multiple clones have been associated with an outbreak, or actually even swarms of closely related organisms that don't form a single clone, but appear through the epidemiological investigation to be involved with the outbreak. And then we always have to ask the question, why is this pathogen on the product? And one of the biggest mysteries in terms of outbreak investigation in my experience was the Salmonella Montevideo outbreak associated with the dried salami product, where the same clone of Salmonella Montevideo was detected in 2 different products from 2 different countries. The likelihood that this same clone would appear would originate from 2 different countries is very low, and so we have to think about how that clone came to be in the 2 different, the black pepper and the red pepper. Also in terms of caveats for whole genome sequencing is the complexity of the data. So the data sets that are produced and how these data are going to be handled in a setting like a public health laboratory. We have issues of quality control that was discussed earlier. And lastly, I'm going to talk a little bit about standardization. There's a lot of methods for data analysis that have been discussed and are being used by regulatory agencies, and there needs to be a process for evaluating those methods, and really for standardization would allow other methods to be recognized. So just from the perspective of FDA, we are moving forward with developing a whole genome sequence-based surveillance of the food supply, and there's a lot of support from very high levels. This surveillance will help strengthen our ability to detect and perform surveillance for outbreaks. It would improve our response to existing threats and also emerging threats. And it's an example of the use of regulatory science to inform how we go about developing standards, performing analyses, and making decisions. I'm just going to provide some examples of, from the last few years of how FDA has been using whole genome sequence to investigate foodborne outbreaks, to conduct traceback, and how it illustrates some of the characteristics that we see as so promising from whole genome sequencing. And I think one of the biggest areas where we find promise is really moving back, rolling back the time where we can identify an outbreak as and the source of that outbreak, and this has been a trend that's been seen over time with the implementation of PFG and PulseNet. That with these new methods, you're seeing smaller and smaller outbreaks, and those are presumably recognized earlier. And this is really a holy grail for regulatory agencies. So it allows us to put our resources into the right places to make the changes that could make an important outcome. So with traditional foodborne illness investigations, we typically had to have a lot of cases to make enough statistical robustness to the data. So in a typical case control study, if you want to identify a specific exposure with illnesses, you would need a large number of cases. And so subsequently, a lot of this work was done after most of the cases were identified. So you would have food entering commerce. The CDC would begin to, and the states would begin to identify illnesses based on a PFG pattern. And then there would be some hypothesis generation, and then FDA, CIFSAN, or ORA, or FSIS would go out in the field and conduct inspections based on those hypotheses, and then you would identify the source. So we're hoping in a future outbreak investigation scenario is that we kind of push that process of hypothesis generation way to the left. That we're able to, because of the robustness of the whole genome data, that we're able to entertain new hypothesis and to begin to collect samples, or perhaps we already have those samples in hand, and that the robust nature of the whole genome sequence data and those matches would impel us at FDA to take a regulatory action. And I'm just going to provide an example of kind of a hint at what we see as the future of this type of new paradigm. This was an outbreak in 2014 involving a soy nut butter that where FDA conducted environmental monitoring of an establishment in February and found 3 positives, and those were submitted for whole genome sequencing, as all Salmonella and Listeria are currently submitted for whole genome sequencing at FDA ORA laboratories. Those isolates matched 6 clinical isolates in 6 different states. I'm sorry, 5 in 5 different states, and then there was a follow-up testing at the same establishment in July, where the additional environmental positives were found. And as shown in this phylogenetic analysis, the isolates from the February environmental investigation formed a very tight cluster. On the top is shown the phylogenetic analysis, and on the bottom just the pairwise SNP distances between outbreak isolates and non-outbreak isolates, and so forming a very tight cluster. This impelled the agency to take regulatory action, to impel the company to perform a product recall, and presumably save lives. We don't know. This isn't a controlled experiment, but we do have information from prior instances where whole genome sequencing was not available. We had another nut butter facility that had what looked like a very long contamination with a salmonella, and there were 42 cases and 10 hospitalizations that were associated with that outbreak. So comparing without whole genome sequencing with whole genome sequencing, a significant number of illnesses and hospitalizations could have been prevented had whole genome sequencing been available earlier. This is another example, not as good. We have Listeria monocytogenes associated with sprouts, also in 2014. So FDA conducted a routine inspection of a facility not soon after a period in which CDC was reporting 5 listeriosis cases in 2 states. 28 samples, including product, food contact surfaces, and processing water, were positive for Listeria monocytogenes. And based on that microbiological result, the manufacturer agreed to a recall. But upon performing whole genome sequencing, first the 5 recent clinical isolates were found to be highly related to the isolates that were obtained through the initial environmental investigation. And then an additional case from 2013 that wasn't in the CDC definition was identified through that process. Subsequently, in October, FDA conducted a follow-up sampling to verify the effectiveness of the company's process, and they found 9 additional samples that tested positive for LM, and those 2 through whole genome sequencing were found to be highly related to the clinical samples, and in the end, in November, the firm closed and ceased production of the mung bean sprout product. So really, what we're seeing here are examples where the availability of whole genome sequencing is moving the investigations to a much tighter. We're finding outbreaks much sooner, and presumably, and I think time will tell, but really based on historical precedent, we'll see smaller and smaller outbreaks, and outbreaks discovered before the major number of illnesses are detected. So I just want to move on to another outbreak that we've investigated. This was a very large outbreak in 2010 involving shell edge. This was the largest product recall in the history of product recalls. And this illustrates the discriminatory power of whole genome sequencing. So this was reported by Dr. Allard and colleagues from our group on a paper that focused on a single PFG pattern that's shown here, PFG pattern 4. This is the most common PFG pattern in Salmonella enteritidis, and is really it's been very difficult to distinguish using both PFGE and other methods distinguished within that pattern. The entire phylogenetic analysis, which included the outbreak isolates from 2010 as well as environmental isolates collected by FDA at the the farms that were identified through traceback investigations, and also historical isolates of the same pattern and different patterns. Identified a number of clades, as shown here, and the two outbreak clades, C1 and C2, had the majority of clinical and also environmental isolates from different farms. And it's very interesting here that the data was able to distinguish very clearly between two major clades within that outbreak, pointing to the possibility of multiple sources of that outbreak. And then if you look at the other data, environmental monitoring also pulled out, as shown in the yellow shade, C6 and C4, that so there were no clinical isolates associated with that, but also found at the same establishment, the same facilities were different other patterns of Salmonella enteritis pattern 4 that had not caused illness, but might eventually have caused illness. So really, what this is showing is that even within a very clonal organism like Salmonella enteritis pattern, PFG pattern 4, you can pick out these smaller groups, and those smaller groups might lead you to products or facilities that you might not ordinarily, we might not be aware of. The next example I want to show really illustrates what we see as a great potential in the whole genome sequence data to demonstrate the geographic signature of Salmonella, such that Salmonella from different parts of the world would look different and have specific sequences that would be indicative of those parts of the world. This is an outbreak of Salmonella bareilles and Ichanga that were associated with a tuna scrape product in 2012. There were 316 cases, mostly in the United States, and the FDA, our group here group in College Park, pulled out clinical and food isolates from the outbreak, and then also picked from our historical collection isolates that had the same PFGE but were not part of the outbreak. And then a number of other isolates that also were not part of the outbreak and didn't have that same PFG pattern. The isolates that with the same PFG pattern came from all over the world, as shown in this figure, including the United States, India, Bangladesh, a number of other countries. There was one isolate from the west coast of India that was the closest match. That was strain 189, and as you can see, so most of the outbreak isolates differed by no more than 5 SNPs within the outbreak clade. The isolate 189 was the closest related to the outbreak clade and differed by 20 isolates. So really pointing out that a common ancestor of 189 in the outbreak lies somewhere in India, on the west coast of India, and really as we began to investigate the Salmonella bareilles 189 came from a shrimp facility that was only 8 kilometers from the facility that processed the tuna samples. There's a work that was published in the Journal of Infectious Diseases recently from Hoffman et al. That goes into this investigation. So GenomeTrakr is FDA's strategy for encouraging the use of whole genome sequencing for food surveillance. Our our overall strategy is that the sequencing, the responsibility for sequencing isolates can be distributed to different laboratories, and this is based on the availability of commercial systems and the reduced cost of sequencing strains. But those labs can build a shared database that would contain draft sequence and minimal metadata, and that would enhance the ability to find similar or matching isolates during outbreak investigations. We believe in using open source data and methods, so provide the methods in an open source way to be transparent, and we're leveraging assets from NCBI and their expertise to store and serve the genomic data. So we think that that all of this data will not only serve very important roles in encouraging partnerships and spur innovation. So this shows our GenomeTrakr laboratories. We work with GenomeTrakr in a lot of different, with states in a lot of different ways, providing assistance that ranges from purchasing reagents to providing labor. We've built a very impressive database of over 12,000 Salmonella and over 3,000 Listeria, along with our partners. And just looking at the Salmonella database, it's a very diverse database that's all publicly available. We're mining this database, and we're encouraging other groups to do the same. This is a presentation at the recent ASM meeting from Gina Ryan and colleagues, looking at the distribution of internal NA sequence variants in the GenomeTrakr database and using the metadata to distribute those into food, environment, and clinical sources. And we're also working with our colleagues at FDA to build real-time whole genome sequencing into the FDA regulatory workflow. So these data from the whole genome sequencing is going into letters and is being used to initiate recalls. We're working on tightening up our methods and our processes so that this data could be used for admissibility as scientific evidence as per the 1993 Daubert decision. And it's shown here that the data the data or techniques should be falsifiable, refutable, and testable, subject to peer review and publication, have a measurable error rate, and have standards and controls, and it should be generally accepted by the scientific community. We published this SNP pipeline, and for the public to use, and that's shown on this slide and can be downloaded. And we're communicating also to the public on how whole genome sequence is being used. Just very briefly, we're working through the ISO, International Standards Organization, on a whole genome sequencing standard for food microbiology. We're looking at varying use cases, so how both regulatory agencies, researchers, and industry could use whole genome sequencing, and that's going to be a basis for our standard. And we're making a lot of progress in terms of defining the scope of this standard in the future. I'm just going to flip through that. In summary, so we have a lot of examples of how whole genome sequencing has defined foodborne outbreaks, where we've sequenced a number of Salmonella and Listeria and closed genomes using our PacBio technology. We're currently sequencing about 600 draft sequences a month within GenomeTrakr. We believe that that environmental sequences are very important and could outweigh the need for extensive clinical monitoring. We believe that the partnership with CDC and USDA and NCBI on real-time tracking of Listeria could be expanded to other pathogens like Salmonella. So, and then just in terms of barriers moving forward, we need continue funding and training to keep moving forward. We need we're finding that a lot of organizations have different data sharing requirements, and this is certainly true on a global scale. We have continual conversations about sharing metadata, and as we're addressing with ISO, we need to address standards in this community so that we understand the methods that we're each using. So again, I think all these presentations, I have a lot of acknowledgements, but it's a very large community of people that are contributing to GenomeTrakr, and I really appreciate that. Thank you.